It is my great pleasure to introduce our speaker for today. Orestis Andriotis is a senior scientist in the biomechanics research unit at the University of Technology in Vienna. He has an undergraduate degree in material science from the University of Patras and a PhD from Southampton University in bioengineering and biomechanics. His research focuses on the development of methods for the nanoscale and microscale characterization of biological tissues, specifically focused on collagen fibrils. And we're really excited to hear about the research, your research today, Orestis. Please go ahead and share your screen.
Thank you very much for the kind introduction, and thanks also to both the Euro-BioImaging and the Austrian Bioimaging Consortium for inviting me to give this presentation in the forum. Our lab is part of the Austrian Bioimaging, and as very well mentioned, we're specialized in multi-scale mechanical characterization of biological samples. And for the presentation today, I decided to focus on the nano and micro scale mechanics, as I think these are two length scales important for cell biology. So now, by training, I'm a material scientist, so I'm not a biologist, and therefore I just show you here a very simplified explanation of the cell-ECM interactions at the nano and micro scale. We know that some cells are embedded in within a matrix made of proteins and fluid, and this matrix is the structural framework for cells to function and reside. Now, as a result of that, cells sense the mechanics of this framework by attaching to several proteins. And in particular, they attach to collagen. And I'm saying this because collagen, as we will see later on, it's one of the most abundant structural proteins found in the human body. Now, you can consider cells to be the biomechanical engineers of this network, and they can intervene to change the mechanics, the structure, and the composition if they deem so. So, for example, if the cells sense that the matrix is too soft, then they might produce more, they get a signal, they might produce more material. Maybe they alter it also chemically to make it again more stiff and come back to a normal state. So this, in fact, process is very important in tissue physiology. In fact, there is a constant degradation and formation of extracellular matrix in a healthy tissue, but sometimes this balance may be altered. And in some of the cases, then it can lead to disease onset and progression in several tissues. This is a classical example of fibrosis, for example, that tissue is formed more than it is being digested, and that can lead to detrimental effects of how the tissue is functioning and may not function anymore, for example. Now, I mentioned collagen, and two of the studies that I will present today focus on collagen. So now, this is, as I mentioned, the protein that cells will attach to eventually and interact with. And just to be a bit of a stereotypical Greek, collagen comes from the Latinized Greek word kolla, which means glue. And the second part of the word is it's gen, which comes from the word ronos, which in this case would mean product. So in fact, it has been used in ancient times to produce glue. And collagen makes up the total collagen family of proteins make up 30% of all the protein mass in the human body. That's quite a substantial amount of proteins. These proteins give the structure and mechanics to biological tissues, and it is the protein that gives also that composes the majority of the extracellular matrix where the cells are residing. Now, where do we find collagen? In fact, it's pretty much in most of the tissues. In fact, it is in all of the tissues that have a in the musculoskeletal system, in joints, in the lungs, in the skin, and composes the connective tissue, in fact. So from that perspective, and all of these tissues, what they have in common is a mechanical functionality. And from that perspective, we can very confident say, or we can see the mechanobiological and biomechanical importance of these tissues and the collagen protein. So the two studies I wanted to show you today, one would be mainly focused on the nanoscale of individual collagen fibrils, and in fact, combined with digestion to see the digestion of these fibrils in situ. And the other study will include also microscale experiments where we look at the mechanics of a fibrotic disease, the idiopathic pulmonary fibrosis. So just before I start with the first study, I would like to very quickly show you something that you probably have already seen in the past, or you know that biological tissues are hierarchically architectured. That means the more we zoom in down to the smallest length scales, we always see some observable structure. And in fact, there are certain collagens that form collagen fibrils. And these fibrils are made of the collagen molecules, which you can see here. I'll just bring up my pointer. One sec. Right, these ultra long collagen molecules, which is about 300 nanometers. What I want you to take out from here is that in a very simplified 2D model, these molecules are staggered in a way, are packed in a way that they give this characteristic pattern when we image in AFM or TEM, for example. Now, as a second part, I would like to mention about these collagen fibrils is that during collagen fibril biosynthesis, these collagen molecules undergo several chemical modifications, and they form crosslinks within the structure. Two types of these crosslinks are enzymatically induced, and a third type is induced by the reaction of sugars with amino acids. Later on, when I will discuss this within one of the studies, I will mainly focus on the enzymatic related crosslinks. Now, one of the things that one needs to do before measuring fibrils at the nanoscale, for that matter, is to be able to get those fibrils somehow on a substrate to measure them. Specifically, we use the atomic force microscope, and we need a stiff substrate for that. And this is done very simple because, for example, if we use tendons are highly abundant in collagen, and with a very minimal, or let's say, with a sample preparation that has no chemicals included, we can very easily dissociate the tendon onto a glass slide and get the fibrils on the glass slide. And the reason why we do that is, if you remember, well, I didn't mention that, I kind of passed it, but well, collagen is a glue-like, it sticks on surfaces. And in fact, it's a very simple way to get this collagen fibril stick on the glass slide that we want to measure. So what you see here is basically an optical microscope image of a piece of tendon that has been smeared out on the glass slide. And if we zoom in one of these fibrils, of these black lines, that is a fibril, we will see this characteristic diperiodicity. Now we use this, and I'm saying this because you need to identify your sample, what is collagen and what is not. So most of what we see here will be collagen, but if we dry it in air and it's dry, we can see this nice pattern, and then we can say with confidence that this is a collagen fibril that we will test. Okay, so moving to the first study, and what we studied here was the digestion of these individual collagen fibrils. Just to remind you, the process of digestion is very important for tissue homeostasis. And if it is impaired, and it is impaired at the collagen, at the level of collagen fibrils, then we would like to know what exactly is happening at this length scale. Of course, digestion is not only important in disease, but it's also in wound healing, or also in diseases like fibrosis. So these mechanisms are very interesting to us. So the protein, well, I did show a very simplified model of how the 2D, the two-dimensional model of how these collagen molecules are staggered. But this is actually not correct, because this is only valid for 2D. This is not the real case. In real case, or let's say in more realistic models, the molecules are not staggered on top of each other, but they're also intertwined around each other. And this we need to know before we start our digestion study, because the way they're staggered and the way they're overlapping on each other will affect how the digestion progresses and our interpretations. Now, the protein we use for digestion is the matrix metalloproteinase 1. It cleaves specifically collagen at the 3/4 of the length from the amine terminus. And this is very important because the amine terminus is located, in fact, periodically found along the fibril. Now, if these lines, the colored lines, are individual molecules in this let's say 3D or 2D to 3D model, shows that this cleavage site is always available for the MMPs to attach. But actually, if we look closely on the cross section of another model of these fibrils, what you see here, these black clouds are individual molecules that are in cross section, meaning they are perpendicular, sort of to say, on your screen. And there's going to be one molecule here, there, and the site at which the MMP will have to bind is hindered by the neighboring molecules. And if we consider the sizes of the enzymes, then in a way, this cleavage site is protected, right? So otherwise, it would just happen spontaneously and go on quickly. But that's not what we observe. So our hypothesis is that this access site is a digestion gatekeeper in a way. So how do we do the experiments? We deposit our collagen fibrils on a glass slide. Then we use atomic force microscopy, which from the last virtual pub, I remember there was given an in-depth on several aspects of atomic force microscopy. In very simple terms, the tip of so we use a force sensor, which is a flexible cantilever. At the end of this flexible cantilever, there is a tip, and we can use the FM and this force sensor to characterize mechanically the sample, and at the same time characterize the topography. Now, if we know how the tip shape looks like, we can also develop contact mechanics models based on the contact between the tip and the sample to find the nano stiffness. What's nice about it is that we can select a region of interest that includes a single fibril, and we can test it in liquid, exposed in the enzymes we want. For every point along the fibril, basically every pixel along this force map, so-called, is a force curve, and we can analyze every force curve to get two information. As I said, we can get the height topography and also the nano stiffness. So what we did, we repeatedly measured digestion in fluid, used activated MMP1. And we did the experiments in a fluid cell that is able to increase the temperature up to 34 degrees during the experiment. And we can now proceed with what we get from these experiments. So on the top, what you see here is a high topography image on the top left. So we can nicely see the individual intact fibrils. I'm saying intact because these, what you see on the background here and also on the right image with a bit more contrast, is a characteristic pattern of a fibril that has been overstrained and damaged. And this gives us some interesting results within this region of interest that we test. So what we see on the, and what is imaged on the top, as it is mentioned here, is the dried state of these fibrils. Right, when we do now these subsequent force maps, high resolution force maps of the individual collagen fibrils, we can observe two things. One, we can see how the height changes during digestion in liquid, and now we can analyze every pixel and see, for example, every force curve and see how the nano stiffness changes. And one of the interesting things that we see is that, for example, for this overstrained fibril, is that it digests or loses height a lot faster than the fibrils that are more intact. And these also lose a bit of height. This is just quantitatively here, but we have analyzed the data as well. And what we also observe is a darkening of this intact fibril, which means it goes from higher stiffness values to lower stiffness values. So somehow there is some dissociation of the fibril that results to the softening of the fibril. Which comes together with height loss. So then what we did, we took those the contact points to measure the height topography and the nano stiffness, and we did this experiment in many different regions of the sample, of course. And now what I would like to show you is some more data, quantitative in that case, how the height changes with respect also to the indentation modulus or the nano stiffness. And what we see here is that if we analyze a number of fibrils, we see that digestion is rather heterogeneous. Some fibrils digest faster than others. In that way, what I mean by digestion is they lose height faster than others. And in fact, others show a different behavior. They kind of swell in the beginning, or the digestion is a lot slower compared to the rest. There's a few examples, but in fact, there are many fibrils that behave like this. It's a matter of fact, you have to think we are only looking now at one very small location, but if we do multiple locations, we see the same effect. And so we have a varying degrees of digestion. Now, we can also quantitatively plot the indentation modulus for every fibril during the experiment. So the darker, in that way, in that case, the darker the bars become, the more the time that the fibrils have been exposed to MMPs. And okay, what you see here as an increase, because maybe that pops out, is just an artifact from the fact that the fibril becomes very small, and we have substrate effects, which is the glass light, which is a lot stiffer than the fibril, and it affects how we get the nano stiffness. Of course, this would be an artifact, but we know that we are in a low height, and that's why we get this increase. But what's interesting is that you can still see the decrease evidently in most of the fibrils, if not all, of the indentation modulus, so the softening in that case. So now the question is what exactly happens, and we looked this into a bit more detail, and what we found is we can see an interesting trend that is worth investigating. And what this trend is the following. If we plot the indentation modulus or the nanostiffness these fibrils have before any exposure to MMPs with the digestion or loss of height after 1 hour, we will notice that there is a trend of stiffer fibrils tend to digest less than softer fibrils. And one thing that we can think of investigating here is, okay, what makes fibrils be softer and stiffer? There are many reasons. One of the simplest way to explain this is the swelling. How much do the fibrils swell in the first place before the digestion happens? But I haven't. This is just the hypothesis now that we led to that we need to test. But what this means in terms of nano stiffness and swelling is that we have seen in other experiments in the past that we know that fibrils swell upon hydration, but you can also, we also do know from these experiments that you can steer the swelling or shrinking. Actually, you can shrink the fibrils using the right medium, and the stiffness of the same fibril will change because of the different swelling or shrinking. And that's simply because if you think about it, these fibrils are made of molecules that are a certain distance apart. And if you change the swelling, then you change the intermolecular spacing. So if you press on something that has more space, in that sense, in between without material or just fluid, then it's going to seem softer than a structure that it's more compact, right? But where digestion comes into place, what we're thinking is that what we want to test is that how the intermolecular spacing may affect the fate and rate of digestion. So if you think that these molecules are further apart, maybe there is more mobility for these molecules also, and then the enzymes can enter the site that these enzymes need to start cleaving the molecules. So there's a number of things that we found out so far. We can determine that the loss of height is a key indicator of enzymatic digestion on individual collagen fibrils. We can confirm there is some, or we can at least assume there is some structural dissociation of the fibrils, because we can see the fibril, while the fibril becomes smaller and smaller, while there is indentation reduction during digestion. Digestion rate varies along fibrils, and it would also be interesting to see why this happens. One way is to test the swelling. And also what's very interesting is that in all of the cases, the overstrained fibrils, of course, I know I showed you only one example, but we have more data on this to support that overstrained fibrils do digest faster. And in fact, this is, if you think about it, this is quite important, because if you have damage in a tissue or overstrained or a tendon, for example, you would need to remove this material and replace it to reestablish the structure and the mechanics, and therefore the function of the tissue. Right. So much about the nanoscale study. And now I'm going to go into the next study, on idiopathic pulmonary fibrosis. So you can think of idiopathic pulmonary fibrosis. Well, idiopathic comes from the fact that no cause has been determined by the doctor that lead to the symptoms the patient have. And pulmonary fibrosis, the fact that the tissue becomes fibrotic. So the hallmark of these pathologies is that there is some inflammation occurring in the beginning that in the epithelium that drives the fibroblasts to differentiate into myofibroblasts, which secrete a lot of extracellular matrix. And eventually what is happening is a progressive fibrosis of the tissue which surrounds the alveoli. The alveoli are the sockets or the sort of the balloons at which, let's say, the oxygen transfer happens to the blood vessels surrounding them. And if you then fill this space with a lot of extracellular matrix, basically what's happening is that that blocks the airways. And if you think this is, let's say a person takes about 29,000 breaths a day, and struggling for each one of them is already too much. In fact, from the time of diagnosis, the median time survival is about 3 years. And so far, there is no drug that can reverse this issue. So what is unclear so far is what the mechanobiological cues that lead to the progression of the disease to the point that the functions don't, the lungs don't function anymore, and in fact patients may undergo transplant. So they might replace basically the defective lung with transplants if that is possible. So the research question we looked at, and this is a study not just, of course, from our laboratory, but this is a collaboration of different research institutions around the world, in fact. And the research question that the leading authors of this work was to find what the specific changes are in the ECM structure, function, and composition that disrupt these mechanohomeostasis of the tissue fibrosis. What we were focused in our lab was to test biopsies and other samples that were provided in terms of micromechanics. We also did some nanomechanics of individual collagen fibrils. And all of these data were then compared with results from analytical chemistry. So for micromechanical characterization, you might have seen this. It has become a quite famous technique. One can use the cantilever to glue microbeads at the end of it, and by microbeads I mean spheres which are made of borosilicate glass, so you need something that it's a lot stiffer than the material you're testing, that are in the size of from 5 to 50 micrometers. It depends a lot on the scale at which you probe the sample. But if you don't use a nano-sized tip and you use a micro-sized tip, then you test a collection of fibrils and a collection of actually the proteins that are in the vicinity of the probe. Again, we can analyze the shape, the exact shape of these beads, and then we can measure in a similar way we did with the nano indentation. We can do location, we can do region of interest, and we can probe in a force map manner. And again, this problem is a contact mechanics problem, which we can analyze and find the micro stiffness. So we tested different number of biopsies and control healthy sample and compared to the control healthy samples. And what we found out is that first, the mean tissue microstiffness is higher in IPF patients or IPF biopsies, and it also varies more in the IPF than in the control healthy samples. And the next question that rises from that is what exactly gives the mechanics at the micro scale? Is it Is it the mechanics of the individual fibrils? Is it compositional changes, or is it also chemical changes at the fibril level? Now, one very interesting result that came out in the very beginning was that there is no change in collagen content whatsoever between the IPF fibrotic tissue and the control healthy tissue. So that was actually a good thing in a way, but it explains a lot. There is not just accumulation of more collagen, but there are other changes that play a role here in driving the stiffness to higher levels in IPF tissue. So we did see, that's from the previous slide, a significant increase in the micro stiffness. But when we tested in the same manner as before the individual fibrils of patients, then there is a significant increase. There is more overlap of the distributions, but there is a significant increase in the fibrils. And something else that I haven't shown here, and but it is included in the paper from Jones and the co-authors, is that the collagen fibrils that come from IPF have a lower diameter overall than the control healthy. So in a way, what happens, if you think about it, is that you have a twofold change at the fibril level. First, you have the same amount of collagen, but you have smaller fibrils, which kind of makes sense. And what smaller fibrils mean is that if you compact them together, they will produce a more dense structure. So in compression, this more dense structure, by just having smaller elements, even if you had the same stiffness at the nanoscale, you would have had a micro stiffness increase. But on top of that, you also have a change of the stiffness of the individual elements that compose the tissue. And now the next question comes, okay, but why do fibrils become more stiff that eventually give the micro stiffness increase? And here is what I want to bring back again, the chemical modification. So one of the things that has been for a long time is the chemical modifications of collagen fibrils. So we saw before that fibrils during formation and maturation, they undergo through several chemical modifications. Two of them is the result of immature and mature crosslinks. And they both come from the reaction of, or the activity of enzymes. So these are enzyme-related crosslinks. And the third one is the advanced glycation end product, kind of an ingenious term for the result of the aging of our tissues, basically, if you think about it. So we didn't look at the sugar-related ones, but we only looked at, so in that study, collaborators looked at the amount of crosslinks in the IPF tissue and compared to control, and they also looked at the activity of these enzymes. So on the left, what you see that we found is that there is a higher ratio of immature to mature crosslinks, which suggests more enzyme activity because these enzymes will act to form the immature crosslinks. So there is more immature to mature crosslinks. And on top of that, the mature crosslinks are also increased overall in IPF compared to control. So then what is to be told here is how can we correlate the mechanics to the enzyme expression or the crosslink addition, basically. And so what we then did, we correlated the micro stiffness with mature crosslinks, and there is we did find a positive correlation there, and at the same time all of the three log cells, so this is the lysyl hydroxylase 3, and that includes also 2 and 4. I haven't included the graphs here. They are all significantly upregulated. So there is a significant activity of these enzymes within the tissue, which means now the cells surrounding the tissue, for some reason, they start producing more of these enzymes. They secrete them, or maybe not. So as I said, I'm not a biologist, so I think this is probably happening in the cell, but I'm not entirely sure. So, but the bottom line is that eventually there is increased activity of these enzymes that will lead to more cross-linking. So the question is, okay, but how can we then inhibit the activity of these enzymes and maybe intervene in a way that we can reduce crosslinks and even reverse them if that is possible? And here's the interesting part. In fact, 2 studies were then done within this. Within this study was to test in vitro a potential selective inhibitor of LOXL2 and 3, and in vitro in both, it was found that this compound reduced the mature crosslinks, as you can see here from the control. And depending on the concentration of this compound, the reduction was also increased of this mature crosslinks. So this is quite a positive outcome. And on top of this, from the same samples that the crosslink analysis was done, some the stiffness or the stress versus strain, what you can see here, the mechanics were also tested for the different samples. So what the samples are, I need to mention that here, is cell spheroids that were produced by culturing IPF cells and healthy controls. So we can now test not at the micro scale, or just above the micro scale in a way, a large collection of these tissues under compression and see the stiffness. And again, there was a 70% stiffness reduction with the drug. There was also an in vivo study on rats that were administered basically a virus that will induce, in a way, the way I understand it, an increase in a factor that increases tissue fibrosis. And then it was given another form of that formula that can be administered orally. And it was also, and what then they tested there, except from the crosslinks after euthanization, then they also tested the lung function in terms of volume and pressure applied. And this came back to normal volumes. So in a way, you can get more air into the lungs using then the drug. So there are some changes happening there. In fact, this then led to the development of a drug that completed phase 1 rather recently, I think, and it's currently undergoing phase 2. So it is potentially a very promising drug that can help with different forms of fibrosis, because we don't find fibrotic tissue only in lungs, but there is liver fibrosis. There is in many different tissues. This can happen. So it seems I'm very early, probably towards the end of my presentation. And with that, I would like to very briefly say a few things of other services except the nano and micromechanics. Well, it's still nano scale mechanics here, but different forms of mechanical tests that we can do. We have, we use mainly atomic force microscopy for imaging, and nanoscale and mechanical characterization by indentation type measurements. All of these instruments are integrated with optical microscopes that with which you can do also fluorescence microscopy, and you can co-register the mechanics with fluorescence if needed. For some of the research questions we cannot unfortunately find off-the-shelf instruments to answer or test our questions, and for that we're also involved a bit in instrumentation, and we have recently developed an instrument with which we can do tensile test of nanofibers, and our test sample, of course, what else would have been the collagen fibrils. But you can also test, we've also tested electrospan fibers, and the versatility there is not a problem. So it's high. Yeah, we can test different types of nanoscale fibers. All of these experiments, of course, in liquid, in the kind of natural or close to natural environment one would expect those samples are exposed to. We also perform microcompression of cell spheroids or cell clusters, and because we figured this can be quite complex deformation scenarios, we also record images or subsequent images during the test, and then we use also mathematical models to implement our own data analysis. Well, of course, I have to say I've been lucky to be working with people with whom I love to work, to ask research questions like this. So I couldn't just say without saying thank you to the people that have contributed to all of the work, or the 2 studies that you have seen, and others within the group for also without the funding partners, this would have never been the possibility. So the project that is related to the digestion of the single fibrils is the WWF, and the one from IPF is the Medical Research Council. And of course, thank you for coming here today, and I'm really happy to answer any questions, and I'm also happy to take any feedback with regards to the presentation. Thank you.